All right, yep, like she said, my name is Will Samuel, and my thesis is entitled When Beavers Get Burned, Do Fish Get Fried? And today we'll be looking at assessing beaver impacts on fish in a fire-dominated ecosystem using eDNA. So kind of starting with the underlying motivation behind my thesis, and that is that the size, frequency, and severity of wildfires is increasing in boreal North America. So you can see the distribution of the boreal biome over here on the right in green, and you can see the distribution of wildfires in Alaska since 1984, that's in orange. Note that those two distributions line up really well, and that's because wildfires are the dominant top-down disturbance process in the boreal biome. Like I said, wildfires are increasing in size, frequency, and severity, and you can really see that illustrated on this chart here on the left, where wildfires have both burned more area and become more frequent since the 1990s. It's also important to recognize that beavers and wildfires have been shown to interact with each other. So a study in 2020 by Emily Fairfax showed that beavers could actually buffer the effects of wildfires on streams, and they basically do this by damming the stream, flooding the riparian zone, and reducing the proximity of that wildfire to the stream channel. Kind of looking at that from the other direction, wildfires have been shown to drive beaver populations. So in this national park, they saw that as the cumulative area burned increased, the beaver population decreased, and a follow-up study 10 years after the fact showed that that population didn't recover, and they attributed that to the high severity of the wildfires, those fires that burn really hot. And both beavers and wildfires affect fish in a variety of numerous and complex ways. And in an ecosystem like I'm going to be talking about today, where beavers are numerous and fires are that dominant process, they have the potential to interact with each other and maybe mediate each other's effects, and even either magnify or reduce each other's effects on fish. So my thesis kind of comes at the center of all of these questions, and I've essentially narrowed it down to one question, which is how do beavers mediate the effects of wildfire on fish? So my first chapter is looking at how do fires affect beavers. I talked about that last year if you were here. And my second chapter is looking at how do beavers affect fish, and I'll be talking about that today. So I'm doing this research in interior Alaska. You can see this gold star is Fairbanks, where we are today. And this is a really large study area, so 21,000 square kilometers. That's larger than the state of Hawaii, Massachusetts, New Jersey, or Connecticut. And about a quarter of this study area has been burned since 1984. Again, I'm just stressing the idea that this is a fire-dominated ecosystem, and all the plants and wildlife, all their distributions are shaped by wildfire. I'd also like to bring up Caribou Poker Creek, which is a long-term ecological monitoring site. This is owned by the University of Alaska. A lot of different ecological monitoring occurs here, but over the past couple of years, my lab has been conducting fish sampling in Caribou Poker Creek, and I'm going to bring this back up later in the presentation. So I just kind of want you to get the idea that Caribou Poker Creek is a good tributary to study that's representative of a lot of other tributaries in the study area. Briefly introducing my study species, Arctic Grayling. It's a widespread fish distributed throughout northern North America. You can see that in purple here. It's mostly used as a sport fish, some subsistence use, and it requires accessibility to headwater and tributary streams, and that's kind of what I'll be discussing today. Briefly introducing one of the tools that I'm going to be using to answer these questions, which is environmental DNA, or eDNA. If you're not familiar with that, eDNA is essentially pieces of DNA that shed from an organism into its environment. So in this case, it's falling off of the fish, either the skin, the fish slime, or it's excreted as fecal matter. And it gets deposited into the environment, and it can persist for some amount of time. The studies show that 24 hours is kind of a minimum, and depending on the conditions, it can persist for a lot longer than that. It's been shown to be transported by flow in streams. The literature shows a maximum of about 1 kilometer. And one of the main limitations to eDNA transport is degradation, which is affected by mostly environmental conditions. So things like the water temperature, the UV radiation that the stream is exposed to, microbial activity in that stream, pH, and a couple unknown factors. And importantly for this project, eDNA has been used to estimate fish abundance and distributions, but it's still a rapidly emerging field. So there's a lot of questions and assumptions that haven't been figured out yet, and that's kind of what I'm going to be talking about today. So this part of my thesis kind of narrows down to two objectives. First, I'm hoping to estimate Arctic grayling distribution and abundance across tributaries in my study area using eDNA. And then once I have those estimates or predictions, I'll compare the results to beaver distributions and wildfire history in my study area. So what that actually looks like as far as the methods, the very first step is to attempt to test the applicability of an abundance model. So here we're trying to see if eDNA is a good representation of fish abundance in streams. So for this, I sampled 10 sites throughout the summer that I'm calling intensive sites. By intensive, I just mean I sampled fish and eDNA, and I sampled them multiple times throughout the summer. So we're trying to use this data to build a relationship between catch per unit effort and eDNA concentration. I also collected eDNA at these other sites that I'm calling broad sites. So here we only sampled eDNA and we only sampled these once throughout the summer. And these are the tributaries throughout my study area that I'm hoping to use that model to predict fish abundance to. As far as the field methods for that, we estimated fish abundance with angling and electrofishing, and then I calculated a catch per unit effort from that. And I also standardized it by the area of the stream that we sampled. And you can see that sometimes it took some creativity and blood, sweat, and tears to get all of our gear out to the sampling sites. As far as the eDNA, we filter, it's basically filtering water. You can see the filters down here on the bottom. We took 3 replicates per site, sometimes more. And we also collected environmental parameters every time we sampled eDNA. So stream velocity, temperature, pH, turbidity, specific conductance, and dissolved oxygen. We folded the filters and stored them in desiccant until we could get back to the lab that day. And then we froze them at -80 until we were ready to send them out to be processed. We processed these at a company called Jonah Ventures, who extracted the eDNA from the filters, and then they incubated or amplified it. And through that process, they can quantify the amount of DNA. So here we're doing a qPCR reaction looking specifically for Arctic Grayling. And then I took those quantitative values that I got back from them and I standardized it by the concentrations. So eDNA per liter. And then I averaged the replicates for every site sampling event. So basically we have an eDNA concentration for every site throughout the study area. Overall, there's a moderate correlation between these data. This is raw data. It looks pretty ugly. Once you log correct both the predictor and response, it makes a much nicer relationship. And we end up with an adjusted R-squared of 0.33. And that includes the habitat covariates, things like water velocity, temperature, pH, which did help explain extra variance. So overall, I'm not totally surprised by these results. I'm not totally surprised that this relationship isn't better. Another study in 2020 by Adam Sepulveda and co-authors attempted to do something very similar, and the results are almost identical to what they did. But I'm not totally discouraged by this because there is this other body of literature that shows that eDNA can be an effective tool for estimating fish abundance. This might just not be the way to do it. So I kind of like to dive into why this relationship between eDNA and catch per unit effort might be so variable. And for that, I'd like to use Caribou Poker Creek as a case study. So here's the fish distribution that my lab observed in 2022. You can see these red marks here indicate the highest extent that we observed of fish last year. And I'd like to focus in on this road accessible area where we sampled eDNA. So here's what the eDNA concentration actually looks like throughout Caribou Poker Creek. So we resampled these sites multiple times. So these are just averaged throughout the whole year. And you can see the eDNA at the top of Caribou Poker Creek, the concentration is pretty low. And as you move downstream, it appears to accumulate or magnify. And this would kind of explain why the modeled relationship isn't that great, because my fish sampling reaches are relatively short compared to what this eDNA might be representing. So the fish sampling reaches were 250 to 500 meters long, and it appears like this eDNA might be representing much more of the tributary than the literature suggests. So you could say that this distribution is just a function of the fish abundance in Caribou Poker Creek. Maybe there's just a lot more fish down at the bottom. But luckily, since we sampled fish in Caribou Poker Creek, we can plot that. So here's the catch per unit effort throughout all the fish sampling sites. And again, this is averaged across all sampling events. And what you can see is that the distribution is pretty uniform. It doesn't seem to change very much between these sites. The one exception is this site over here on Caribou Creek. And that's kind of a unique spot where the fish actually get piled up right beneath the beaver dam that they can't pass over. And so they're really concentrated in this pool, really easy to catch. So that catch per unit effort value might be partially artificially inflated, but I also think that high fish concentration is represented because this eDNA concentration in Caribou Creek, it's kind of hard to see here, but it's about twice that of what's in Poker Creek. So kind of thinking to those assumptions that I introduced the presentation with, the idea that eDNA can persist for 24 hours or so in a stream environment. And the literature says that it can only travel about 1 kilometer in a stream. And we can, now that we have some data, we can kind of test those ideas. So we know where the most upstream fish is, or have a rough idea of that. And so that's this gold star red line here. That's about 8 kilometers or 8,000 meters along longitudinally along this stream to the bottom eDNA reach. And while we were sampling eDNA, we also collected velocity. So we know averaged across the whole year, all the sites and sampling events, we know that kind of the average stream velocity. So we have a distance and a rate. So we should be able to calculate how long it should take that eDNA to move from the top of the tributary to our bottom eDNA sampling site. And when you do that math, it comes out to about 392 minutes or 6.5 hours. So this is kind of a rough estimate, but it's a good test to check our assumptions. And it's a good test to check our assumptions. And it shows that this first assumption that we're assuming that we're sampling within that 24-hour mark is most likely true. And this other assumption that the eDNA travels 1 kilometer or less, I think I'd like to update it and say that as long as that eDNA doesn't settle or degrade in the stream within 6.5 hours, or maybe 8 or 10 hours in a longer tributary, that it can travel much further than this previous literature suggests, maybe 5 or 10 kilometers in a stream. And I think that the reason for that is because of our study area. We kind of live in this ice box in the north, and a lot of eDNA research hasn't been conducted up here yet. So I think the degradation rate is likely a lot slower here than in these places where the literature has been established. Our streams are colder, they're less productive, so there's less microbial action, and we have much lower UV radiation because the sun's at a lower angle and has to travel through more atmosphere before reaching our streams. So this is kind of just this case study, and it's kind of observational. I don't know if I'm going to have the data to prove this, but it is kind of a good way to validate the next step of my project, which is to predict this fish abundance or estimate it across these 56 tributaries. So to do that, I'm comparing a couple different model structures and techniques. I kind of already talked about the linear model, and that may or may not be a good method to estimate fish abundance here. There's other model structures that we can use, something like an N mixture model or a multi-state occupancy model, which would kind of fit the eDNA data set a little better. And there's also potential to maybe incorporate allometric scaling using a Bayesian context. Overall, I'm not totally sure what I'm going to do here, but I think in an ideal world, I'd be able to try a couple different model structures and use those to predict out across my whole study area. And then compare the results from those. And if the general trends of those multiple models are all similar, I think we can have pretty high confidence in our results. Once I have those predictions made, I'd like to summarize the habitat factors that are associated with that. So for my first chapter, I summarized the beaver distribution throughout my study area. You can see those black points here are beaver ponds and the wildfire history as well. So we can see if streams with a lot of grayling or few grayling have high beaver density or low beaver density, and whether they were burned recently or not. So with that, I'd like to thank everyone who contributed to this project, my committee and co-authors, my technicians last spring and summer, the volunteers who helped me with fieldwork, and I really couldn't have completed this project without them. And I'd like to thank funding from the Department of Defense and Alaska EBSCOR program. With that, I'd be happy to take any questions.
Yeah, when I was looking at your maps of like the eDNA density and the density of grayling you sampled, it looked like, yeah, perfect. The eDNA like really kind of amplifies downstream. And you took stream velocity.
Mm-hmm.
Okay, it'd be cool if you could account for the discharge somehow and kind of get the water volume reflected, but something you could do after the fact is try incorporating stream order. It looks like you only have a couple there, but that could kind of get at that kind of effect of more tributaries coming in and that bio concentration of that eDNA.
Yeah, that's a great point. Yeah, I haven't really looked into that yet, but it's definitely something I'll consider in the future. The fish catch green effort should account for that, since that's standardized by the area of the stream. The eDNA is a concentration, so it should kind of already be standardized by that, but I think it's worth looking into for sure. Joe?
Did you guys take eDNA above the barriers that are on that map?
Yeah.
To see if there was eDNA present in the water.
Yeah,
above the distribution?
yeah, so I didn't have a chance to talk about it today. That was the other thing that I did all summer long. I had actually 6 other sites beyond this, what I talked about today. And yeah, so maybe another time when we have time, I could show you that. And basically what I saw was that for the most part, when we didn't observe fish above these beaver dams, we also didn't observe DNA, or if we did, it was really, really low. So it seems like they generally agree. We're still kind of playing with that component though. Questions from Juno or online?
You have a question here in Juno from Megan.
Yeah, nice talk. I was just wondering if you think that the relationship between might differ between burned and non-burned reaches. I was just thinking about the effect of UV, or do you think just temperature is enough to capture that?
Yeah, that's a good question. I haven't really looked into that. I think if I could incorporate that, it would probably be in this next step when I'm predicting out. Because we have a much bigger data set there and you know, we have more variation between the burned and unburned. As far as these broad sites or these intensive sites, these red squares that I tried to build my model with, I didn't really set that up in a framework to compare burned versus unburned. But we could definitely pull that out of this broader data set. So once I move to that, it's something we'll be looking at.